Hello everyone, my name is Dhruvin Patel and today I would talk about research on enhancing safety of drinking water. Water is crucial in maintaining normal physiological and biochemical function of the body. However, contaminated water with pathogens like viruses, protozoa, bacteria causes wide range of diseases, which often infects the small intestine and leads to diarrheal diseases. Water disinfection process have been introduced to alleviate this problem, and it includes physical method like sedimentation and filtration, chemical use of chemical disinfectant to control the pathogens, and other methods like UV rays and heating. This chemical disinfectant oxidizes the organic matter and bromide present from the source water to form disinfectant byproduct DPPs. Chlorination, which was the first pioneer disinfectant, forms chloroform and other trihalomethanes, which are now regulated due to their abundance and carcinogenic effects. Only 12 of 700 identified DPPs are regulated globally. When chloramine is used as a secondary disinfectant, nitrogenous disinfectant products and DPPs are formed. In vivo studies have found exposure to HANs, which are still unregulated DBPs, cause changes in DNA in CHO, DNA alkylation, and mutagenic properties in bacteria. However, in vitro toxicity assays, which are required to completely elucidate their effects, are not yet developed. The objective of my project was to evaluate the stability of DHANs, and specifically TBAN and TCAN, in cell buffer, and also in cell culture media using SPME GCMSMS. In media, we use the Eagle cell culture media and a phosphate carbonate buffer at pH 7.4. This is just the structure of the compounds being studied in this project. SPME sampling technique was used, in which basically the injector was pierced in the vial. The samples were collected on the fiber, and the analytes were loaded on the GC column. In GCMSMS, and particularly in GC, the instrument parameters are set so that only the compounds of interest are allowed to reach the detector after passing through the separation column. Once in the Tandem mass spec, the analytes are fragmented, ionized, such that only the ions with specific charge, mass to charge ratio, are allowed to reach the detector. This allows a detection at high precision and accuracy. We spent a lot of time optimizing the method, and we found the collision energy of 15 electrovolts was optimum, and these are the MRM transition used. This plot shows the ratio of area under TCAN and ISTD on this axis, and the same for TBAN on this axis as a function of time in hours. Right at the top, it can be seen that TBAN was very unstable than TCAN since day 0, as it is all represented by a very different drastic scale. And TBAN completely degrades after 24 hours and is basically not present in the media. TCAN decreases by 90% after the first 24 hours, reaching 0.02 from 0.017, and more or less stabilizes after 48 hours. Similar trends were observed for TBAN as well as TCAN in the physiological buffer, which was at pH 7.4. This plot shows the stability of TCAN and TBAN in buffer and media. TCAN has as seen here on the left plot, TCAN has an interesting stability pattern where it was very abundant in the media at 0 hours, but degrades very fast and reaches kind of similar level after 24 hours. After 48 hours, it was actually more stable in the buffer, and after 72 hours, it was more or less similarly stable in both the treatments. However, TBAN was clearly more stable in the buffer, but it degrades after 24 hours in both the cases. This means that TBAN is more unstable than TCAN overall. Applications of this research are directly applied in developments of toxicity assay to investigate biological effects of DHANs for global regulation, and also increasing the safety of drinking water. And these are the references.